Welcome to the Deep Dive, the place where we grab some really ambitious, sometimes seriously complex research papers and, well, pull out the key insights for you.
And today we've got a topic it's genuinely ambitious. We're trying to connect the dots between mycology, that's fungi quantum physics, the really deep theoretical stuff, and cutting-edge supercomputing.
Yeah, it sounds like three completely separate worlds.
It really does. And the source material reflects that. We're looking at everything from, high-level quantum computation theory papers to really dense biological studies filled with complex math like partial differential equations and detailed molecular dynamics. It's a unique mix.
So our mission today is to unpack this stack of sources. We want to explore claims about things like a quantum computer made from mushrooms.
Yeah, really. And even look at how quantum algorithms are being designed to fundamentally optimize how plants suck up nutrients.
It's about spotting this new frontier, potentially, in sustainable tech and computational biology. We're connecting the dots from nanoscale biology all the way up to the quantum software needed to understand it. It's quite a synthesis, moving from, say, a theoretical biocomputer concept from a group called Quantum Creative Solutions right down to how individual protons might tunnel through a fungal membrane.
And then back up to the quantum algorithms needed to model and maybe even predict agricultural outcomes. The scope is just huge.
Okay, so let's start with that hook, the really eye-catching idea, this mycelium quantum computer, the MQC, the idea of a living computer, maybe even self-repairing. It sounds like pure sci-fi.
It does, but the research grounding is in the unique properties of fungi, specifically the mycelial network.
So Quantum Creative Solutions, they're proposing this MQC, and it's not just sticking silicon onto mushrooms, right?
No, not at all. The claim is much deeper. They're arguing that the mycelium network itself, its structure, its chemistry, inherently provides the right conditions for quantum computation. Things like stability, noise reduction, maybe even fault tolerance.
Which would be a huge shift from how we build computers now, the incredibly energy-hungry, resource-intensive manufacturing.
Exactly. It's a fundamentally different approach, leveraging biology instead of fighting against its complexities.
All right, let's unpack that first big piece. I mean, usually quantum computing means ultra-cold labs, massive shielding. So why fungi? How does this web-like mycelium network, these tiny hyphae filaments, possibly provide a stable base for quantum hardware?
Well, the sources suggest the mycelium tackles two massive headaches in quantum computing. First is stability and, crucially, noise reduction.
Right, quantum states are ridiculously sensitive. Decoherence, the collapse of the quantum state, is the big enemy.
The absolute killer. Any tiny vibration, heat fluctuation, stray magnetic field, poof, it's gone. But the idea here is that the mycelium structure, its complex fractal geometry, the density of the network acts like a natural shield.
A shield, how?
The physical structure, combined with the sort of molecular rigidity of the fungal cell walls, creates this remarkably stable microenvironment. And the way the network branches, which evolved for efficient nutrient transport, might naturally dampen external noise, heat, vibrations.
So it protects the quantum states, keeps them coherent longer.
That's the hypothesis.
Oh.
Essentially, the structure itself performs a function. We currently spend, millions, billions on cryogenics, massive shielded rooms.
Okay, so the network structure is doing the job of a high-tech lab facility, in a way.
That's the argument. And the second factor is communication efficiency. Quantum computing isn't just about holding a state. It's about entanglement, processing information across qubits. The mycelium network, already optimized by evolution for rapidly moving resources around, potentially offers these incredibly efficient pathways for quantum information to travel and be processed within this biohybrid system.
That's fascinating on the structural side, but stability and communication pipes aren't enough. You still need the actual qubit, right? The thing doing the computing. If it's not a superconductor or a trapped ion, what is it in the MQC?
This is maybe the most intriguing part. The sources point to leveraging nature's quantum dots.
Naturally occurring quantum dots.
Apparently, yes. Certain nanoscale structures naturally present within the mycelium. Could be metabolic byproducts, specific molecular complexes. The sources aren't entirely specific on the exact identity yet, but these structures function like synthetic quantum dots.
Meaning they act like tiny traps.
Tiny self-assembled traps for individual electrons. And it's the quantum properties of those trapped electrons, their spin, their ability to be in superposition that would be harnessed for computation.
Wow. So the fungus itself essentially manufactures the core processing units as part of its life cycle.
That's the core claim. And if true, it sidesteps one of the biggest hurdles in quantum hardware manufacturing.
Right, the need for those billion-dollar clean rooms, the incredibly precise fabrication.
If the components are biologically inherent, self-assembling, well, you eliminate that massive cost and complexity. It's not just cheaper, it's a philosophical leap towards sustainable computing.
Lower energy footprint, fewer exotic materials.
Definitely. Fungi don't need a dedicated power plant like some supercomputing centers do.
Okay, but let me push on this living computer idea. Biology is messy, right? Isn't there a risk of biological noise, metabolic waste, humidity changes, things dying off? Couldn't that just crash the whole system?
That's a really important question, and the sources do address it. They frame the living aspect not as a weakness, but maybe its greatest strength.
So,
They talk about resilience and adaptability. The idea is that the biological parts don't just compute, they also give the system inherent self-repair and maybe even self-replication capabilities.
self-repair. You mean like if a bit of the network gets damaged or a qubit decoheres, it can just grow a new pathway, reroute the calculation.
That seems to be the implication. A fault-tolerant network that heals, much like a living organism. And self-replication that suggests the system could potentially scale itself up. Need more processing power. It doesn't wait for a factory upgrade. It uses its own growth mechanisms.
A self-optimizing system that improves over time.
That's the vision laid out. A level of resilience and autonomy that current silicon tech just can't match.
Okay, that MQC concept is, well, it's mind-bending. A sustainable, self-healing quantum computer. But to actually prove fungi operate with that kind of quantum efficiency, you need incredibly precise data, right? Down at the molecular level.
Absolutely. And that brings us neatly to the second major part of our sources, mycoponics.
Before we get into the lab setup, maybe quickly remind us why these fungal networks are so important out there in the real world, in nature.
Right. So beneath forests, fields, almost everywhere, you have these mycorrhizal networks. Specific fungi, like AMF or Ectomycorrhizae, forming these vast underground partnerships with plant roots.
The Wood Wide Web idea, essentially.
Pretty much, they act like extensions of the plant's root system, dramatically boosting the plant's ability to grab essential nutrients, especially things like phosphorus and nitrogen, which are often locked up in the soil.
So they're crucial for ecosystems.
Absolutely critical. They enhance forest resilience, help sequester carbon in the soil, and, importantly for us today, they reduce the need for chemical fertilizers in agriculture because they make plants so much better at feeding themselves.
But studying how they do that so efficiently is tough in actual soil. It's messy, full of other microbes.
Exactly. Soil is just too complex, too heterogeneous. If you want to isolate the specific fungus-plant interaction and look for subtle, perhaps quantum effects, that's the problem. Mycoponics, developed by Dr. D. Marshall Porterfield's group, was designed to solve.
So what is mycoponics?
It's a really clever soil-less cultivation system. Instead of soil, they use a sterile, transparent hydrogel substrate.
Like a clear jelly.
Sort of, yeah.
Yeah.
The beauty is it lets them study just the fungus and the plant root interacting. Without all the confounding factors of soil, it gives them unprecedented control and visibility.
They turn a messy field problem into a clean, controlled lab experiment you can literally watch. What kind of specific setups do the sources describe?
They detail some pretty rigorous protocols. They use specific, well-characterized fungi, often things like Glomus enteratices, known for being great at phosphorus uptake, or Rhizophagus irregularis for both N and P.
And they pair these with standard crops.
Yeah, typically maize or wheat, plants known to form strong symbiotic relationships. The whole setup is designed for maximum precision.
How do they make sure it's still relevant to nature if it's so sterile and controlled?
They incorporate lab-on-a-chip technology within the hydrogel. These microfluidic chips let them create precise gradients of nutrients, mimicking the patchy distribution you find in real soil. So the fungus still has to work to scavenge efficiently, but now every variable can be measured.
Okay, now the really tricky part, tracking the nutrients. How do you actually see and quantify phosphorus moving from the gel through the fungus into the root, possibly at the single molecule level? Sounds like you need a whole toolkit.
It's definitely a multimodal approach, combining chemistry, physics, advanced imaging. The sources lay out about 3 main strategies.
What's the first?
First is good old isotope tracing. They add nutrients labeled with specific isotopes like nitrogen-15 or phosphorus-32, which are heavier than the common forms.
So you can track them specifically.
Exactly. After the experiment runs, they harvest the plant and fungal bits and use high-resolution isotope ratio mass spectrometry, IRMS, to measure exactly how much of that labeled N or P made it into the tissues. It gives you the final quantitative bottom line on transfer.
Okay, that shows you how much got transferred. But phosphorus chemistry is complex, right?
Yeah.
Does its form change during transport?
That's a key question. And for that, they use nuclear magnetic resonance, NMR, spectroscopy. NMR is brilliant because it can distinguish between different chemical forms of phosphorus orthophosphate, pyrophosphate, other organic phosphate compounds. It reveals the chemical journey of the phosphorus molecule.
Right, so IRMS for quantity, NMR for chemical identity. But neither shows you the movement in real time, the dynamics.
Correct. For the real-time visualization, they need something more dynamic. And that's where quantum dots, QDs, come in as markers.
Ah, back to quantum dots, but used differently here.
Yes, here they're used as tiny fluorescent tags. They chemically attach these QDs to the nitrogen or phosphorus molecules. When you shine light on them, they glow a specific color, depending on their size.
So you can literally watch the glowing nutrient molecules moving.
That's the goal. But visualizing them deep inside the hydrogel and the fine fungal hyphae requires specialized microscopes.
What kind?
They mention standard confocal microscopy for high-res 3D images near the surface. But crucially, they rely heavily on two-photon excitation microscopy, or TPEF.
Why two-photon?
TPEF uses longer wavelength light. This means less scattering as the light goes through the tissue, allowing you to see much deeper. And very importantly, it causes less photo damage, less risk of cooking the living cells you're trying to observe.
So you get deep imaging without killing the experiment. Makes sense. The sources also mention fluorescence lifetime imaging microscopy, FLIM. What does that add?
FLIM is really sophisticated. Instead of just measuring where the light is, it measures how long the fluorescence lasts after the QD has excited the fluorescence lifetime. This lifetime is incredibly sensitive to the QD's immediate molecular environment.
Like the pH, or if it's bound to a protein.
Exactly. Changes in pH, viscosity, binding events, they all affect the fluorescence lifetime. So FLIM gives you dynamic information about what the nutrient molecule is actually doing and interacting with as it moves, not just where it is.
Okay, that's an incredible amount of data blinking lights from QDs moving in 3D space over time with lifetime information layered on top. How did they translate that flickering into hard numbers, like how fast the nutrients are moving?
That requires some serious statistical analysis using techniques like image correlation spectroscopy, ICS.
ICS, what's the basic idea?
Okay, imagine watching a single spot on the fungal hypha and seeing these tiny QD lights flash as they pass through. ICS analyzes the fluctuations in that light signal over time and space.
So if the flashes are quick and brief, it means fast movement.
Pretty much. If a QD zips through your observation spot quickly, the fluctuation is sharp. If it lingers and diffuses around, the fluctuation is broader. By mathematically analyzing these fluctuations, calculating things called autocorrelation and cross-correlation functions, ICS can extract quantitative values like the average diffusion coefficients and transport velocities of the nutrients.
Wow, so they can actually measure the speed of nutrient packets moving along the fungal highway.
At the single molecule level, yes. And it's this level of detailed quantitative data showing incredibly high efficiency, especially under stress when nutrients are scarce, backed by complex mathematical models of diffusion and uptake that really pushes the boundary.
It suggests that just standard biochemical explanations might not be enough.
Exactly. The observed efficiency is so optimized, so robust, it strongly hints that classical mechanisms alone don't tell the whole story. It practically screams for a quantum explanation.
Okay, this is a really key moment in the discussion. We're shifting from observing this incredible biological efficiency in fungi to asking why it's so efficient. What's the quantum physics actually doing down there?
The need to invoke quantum mechanics really comes into focus when you look at processes involving very light particles, electrons, and especially protons and energy barriers. Classically, you need enough energy to get over a barrier.
Like pushing a ball up a hill.
Right. But in biological systems, we often see transfers happening much faster and more efficiently than classical physics would predict, especially when there isn't seemingly enough thermal energy available.
So let's start with the most famous quantum weirdness, quantum tunneling. How does this help, say, a proton move across a fungal membrane to drive nutrient uptake?
Quantum tunneling is this bizarre consequence of particles also behaving like waves. It means there's a non-zero chance a particle can just appear on the other side of an energy barrier, even if it doesn't have enough energy to climb over it. It tunnels through.
It cheats the hill.
It completely cheats the hill. And the sources confirm this is likely crucial for rapid proton movement across fungal membranes. These proton movements are fundamental. They create the electrochemical gradients that power many nutrient transport systems. Tunneling provides a super fast shortcut.
Instead of the cell spending lots of energy to pump the proton over, it just tunnels.
It significantly increases the probability of crossing, especially across the short distances inside proteins or across membranes. We can even estimate the probability using math like the WKB approximation. This shows the tunneling rate depends heavily on the barrier's height and, crucially, its width. Narrow barriers facilitate tunneling,
Suggesting maybe fungal membranes or transport proteins are structured to create these narrow barriers.
That's a fascinating possibility. And it's not just protons. Tunneling is also vital for electron transfer in redox reactions, fundamental energy conversion processes, like in respiration involving cytochrome proteins. The speed of these electron transfers is often modeled by Marcus theory, which is itself deeply rooted in quantum mechanics. It shows these basic life processes rely on particles taking these non-classical shortcuts.
Okay, tunneling helps particles get through barriers. But the sources also talk about quantum coherence, improving efficiency. How does coherence, this idea of superposition, help transport?
Coherence is about maintaining that wave-like nature. A coherent particle, like an electron or proton involved in transport, can effectively explore multiple possible pathways simultaneously.
Like it exists in multiple places at once.
In a probabilistic sense, yes. It's like a wave splitting and exploring different routes. And just like waves, these different pathway possibilities can interfere with each other.
Constructive and destructive interference.
Exactly. If the pathways interfere constructively, it dramatically boosts the probability of the particle reaching its target destination quickly and efficiently. It finds the optimal path by effectively exploring all paths at once. We see similar mechanisms proposed for the incredible efficiency of photosynthesis.
So the fungal system might be using coherence to run this constant internal quantum search for the fastest nutrient transport route.
That's the hypothesis. The overall efficiency, let's call it 8-8I, in a coherent system, is directly linked to quantum properties, like the energy difference between the start and end states. Better management of the quantum coherence leads directly to better biological performance.
It sounds almost too perfect.
Yeah.
A perfectly efficient quantum machine running inside a fungus. But biology happens in warm, wet, messy environments. What about decoherence? Doesn't the environment just destroy this beautiful quantum state?
Ah, yes. Decoherence is the unavoidable reality check. In any real biological system, the quantum state is constantly interacting with its surroundings, water molecules bumping into it, thermal vibrations.
Noise.
Exactly. This environmental noise disrupts the delicate superposition, causing the coherence to be lost very, very quickly. The particle gets forced out of its wave-like superposition and snaps into a single classical state.
So the quantum advantage is fleeting.
Extremely fleeting, potentially on the femtosecond or picosecond timescale in many cases. Researchers model this breakdown using tools like the Lindblad master equation.
What does that equation tell us?
It mathematically describes how the quantum state, represented by something called the density matrix, evolves over time when it's coupled to a noisy environment. It accounts for these dissipative effects and shows how the system inevitably drifts from a pure quantum state towards a mixed, thermal, essentially classical state.
So it predicts how quickly the quantum magic fades.
Yes, and it shows that the rate of fading is strongly dependent on temperature. The warmer the environment, the faster the decoherence. The system is always racing against this environmental decoherence.
So the biological strategy might be to use these quantum effects, tunneling, coherence, for brief, ultra-fast steps, accomplishing something vital before decoherence takes over.
That's a very likely scenario. It's about exploiting the quantum advantage in that tiny window before the environment thermalizes everything back to classical behavior. The maximum potential efficiency is quantum, but the realized efficiency is limited by how long coherence can be maintained against decoherence.
Okay, so we've established that fungi likely are using quantum mechanics for efficiency. This is amazing. So the next big question is, Can we use quantum computers to figure out how to make these processes even better? Can we use quantum algorithms to optimize these biological systems, maybe for agriculture?
Exactly. We shift now from quantum effects in biology to using quantum computation for biology. And the reason we need quantum computers is the sheer complexity. Many core biological processes are what we call combinatorial optimization problems.
Like protein folding.
Protein folding is the classic example. A protein is a long chain of amino acids, and it needs to fold into a very specific 3D shape to function. Finding that one correct lowest energy shape out of a staggeringly huge number of possibilities.
The search space is astronomical.
Exponentially large. For even a moderately sized protein, a classical computer trying to brute force check every possible fold would take longer than the age of the universe.
So classical computers hit a wall. How do quantum algorithms help? The sources focus on two main ones. Let's start with the variational quantum eigensolver, or VQE.
Right, VQE is a really important algorithm, especially in this near-term era of quantum computing. It's a hybrid algorithm.
Meaning it uses both quantum and classical computers together.
Exactly. The goal of VQE is to find the minimum energy state, the ground state of a molecule, like one of those nutrient transport proteins. First, you map the molecule's physics, all the interactions between its atoms, onto a mathematical description called a Hamiltonian that the quantum computer can understand.
Okay.
Then the quantum computer prepares a guess, a trial quantum state based on some parameters. It measures the energy of this state according to the Hamiltonian. Now, a fundamental rule called the variational principle guarantees that this measured energy will always be greater than or equal to the true ground state energy.
So you know you haven't undershot the lowest possible energy.
Precisely. The classical computer then takes that energy measurement, runs an optimization routine, and tells the quantum computer how to adjust the parameters to prepare a new trial state that should have lower energy. You repeat this loop: prepare state, measure energy, classically optimize parameters, iteratively refining the state until you converge on the minimum energy.
And for our fungi, finding that minimum energy state for a transport protein tells you its functional shape.
Yes, because the protein's function is dictated by its 3D structure, which corresponds to that lowest energy configuration. VQE helps us find that structure.
Okay, that's VQE. The sources also talk a lot about the quantum approximate optimization algorithm, QAOA. How is that different?
QAOA is also often used for optimization, and it can be applied to problems like protein folding too, but its approach is a bit different. It's generally designed for broader combinatorial optimization problems, finding the best solution among many discrete possibilities, not just finding the lowest energy of a molecule.
So maybe more versatile.
In some ways, QAOA works by setting up two Hamiltonians. One is the problem Hamiltonian, HPLA, which encodes the problem you're trying to solve, like the protein energy landscape. The other is a mixer Hamiltonian, which helps the algorithm explore different possible solutions.
And how does it use them?
It applies these Hamiltonians in an alternating sequence for specific durations controlled by optimized parameters, gamma and beta. You basically evolve the quantum state back and forth between exploring the solution space via H-infinity and being guided by the problem's constraints via HTOF.
And the more steps you take, the better the answer?
Generally, yes. As you increase the number of alternating steps, the circuit depth, the final state gets closer to the optimal solution, the one that minimizes the problem Hamiltonian. QAOA is often seen as good for finding very good approximate solutions relatively quickly, especially for really large problems where finding the absolute exact minimum might be too hard even for VQE.
So VQE aims for the exact ground state energy. QAOA is more about efficiently finding near-optimal configurations in vast search spaces.
That's a good way to put it. They're both powerful tools suited for slightly different nuances of biological optimization problems.
Now, a reality check. Current quantum computers are still relatively small and noisy, right? The NISQ era noisy intermediate scale quantum.
Yeah.
We can't just simulate an entire fungal cell membrane quantum mechanically?
Absolutely not. That's computationally infeasible right now, which is why the sources emphasize these hybrid classical quantum molecular dynamics, MD, simulations.
Combining the strengths of both worlds again.
Exactly. Classical molecular dynamics, MD, uses simplified models, called force fields, to simulate the large-scale movement and behavior of molecules over time. It's great for simulating how, say, A nutrient diffuses through the bulk of a membrane over nanoseconds or microseconds.
It misses the quantum details.
Right. The force fields are approximations. They don't capture the quantum effects like tunneling or precise bond breaking forming during a reaction. So the hybrid approach is smart. You use classical MD for the bulk of the system, the parts that are moving around but not undergoing complex quantum changes.
And save the quantum firepower for where it matters most.
Precisely. You integrate highly accurate quantum electronic structure calculations, maybe using VQE, or another method like density functional theory, DFT, only for the small critical region, like the active site of a transport protein.
Because that's where the actual nutrient binding, the proton tunneling, the electron transfers are happening.
Exactly. That's where the quantum mechanics are dominant. By focusing the expensive quantum calculation just on that small crucial zone, you get the quantum accuracy where you need it, while the classical MD handles the overall dynamics efficiently. It's the most practical way forward currently.
Okay, so you've got experiments, mycoponics, generating data, and hybrid simulations, MD plus VQ, EQ, AOA, making predictions. How do you close the loop? How do you use all this information effectively? That leads to data integration and machine learning.
Yes, this whole research effort is envisioned as a continuous feedback cycle. The experimental data from mycoponics nutrient uptake rates, environmental conditions like pH, temperature flows into the system. Simultaneously, the quantum simulations provide theoretical data calculated protein binding energies, predicted folding structures, optimal reaction pathways.
And machine learning helps make sense of it all.
Right. You have this incredibly diverse data stream, some from real-world measurements, some from quantum calculations. Hybrid machine learning models are used to fuse this information.
What does hybrid mean in the ML context here?
It often means using classical machine learning architectures, like neural networks, but augmenting them with quantum-inspired or actual quantum components. The sources mention things like quantum kernel estimation, QKE, or variational quantum classifiers, VQC.
Quantum kernel estimation, what does that do?
QKE is interesting. It basically takes your complex classical data points, like the combined experimental and simulation results, and maps them into a much higher dimensional space, using quantum principles, a quantum feature space. In this space, complex patterns and nonlinear relationships that might be hidden in the original data can become much easier for the machine learning model to identify.
So it helps find subtle links between, say, soil pH and quantum tunneling rates that a standard model might miss.
Potentially, yes. The overall goal of this integrated ML system is to build predictive models. Models that can say, under these specific environmental conditions, this particular strain of fungus will be most effective. Or, to maximize phosphorus uptake, you should aim for this pH and temperature profile.
Turning all that complex science into actionable insights for agriculture or biotechnology.
Exactly. Guiding experiments, selecting better fungal inoculants, optimizing conditions in real time based on this constant flow of data and simulation. So if we zoom out from just the fungi, this whole approach using quantum insights and quantum computation to understand and optimize biology has really broad implications across the life sciences. The fundamental advantage of quantum computing comes down to how it handles probability, using complex amplitudes.
Which allows for interference, constructive and destructive between different computational paths, something classical bits can't do.
Right. And that interference is the source of quantum speedup. We generally think of two main types. First, there's exponential speedup.
The holy grail, like Shor's algorithm for factoring large numbers.
Exactly. In biology and chemistry, this is most relevant for simulating quantum systems themselves, molecules, with lots of strongly interacting electrons, like complex catalysts or drug molecules. Classical computers just choke on the complexity. Quantum computers promise to handle these simulations exponentially faster.
And the second type of speedup
is polynomial speedup, often quadratic, like Grover's algorithm for searching unsorted databases. Searching n items classically takes about n steps. Grover's does it in roughly the square root of n steps.
Still a huge advantage for very large n.
A massive advantage. So where else in biology might these speedups apply? Well, take genomics.
Dealing with massive amounts of sequence data.
Huge amounts. Quantum algorithms are being developed to improve tasks like sequence alignment and matching. Things like hidden quantum Markov models, HQMMs, might process sequence data in superposition, potentially offering faster or more accurate alignments than classical HMMs.
And genetics involves complex relationships, right?
Definitely. Problems like figuring out ancestral recombination graphs, ARGs, mapping the history of how genomes recombine over generations, are incredibly complex optimization problems on graphs. Quantum approaches like quantum annealing are being explored as potential heuristics to tackle these classically hard problems.
Okay, what about functional genomics? Understanding what all those genes do, how they interact.
That often involves analyzing massive data sets like gene expression levels across thousands of genes. Building models like Bayesian networks to capture these interactions or performing complex clustering is often computationally intractable classically.
So quantum heuristics again?
Yes, quantum approaches using things like quantum Boltzmann machines, QBMs, or quantum variational autoencoders, QVAEs, might offer advantages. QBMs, for example, could potentially sample from the complex probability distributions described by this high-dimensional gene expression data much more efficiently than classical methods, revealing hidden regulatory networks.
And then there's the ultimate biological complexity, neuroscience. The sources call it a multi-scale challenge.
It really is. You're trying to connect everything from molecules and synapses up to large-scale brain activity, like in fMRI scans, and complex behavior. The data is incredibly high-dimensional and nonlinear.
Can quantum computing help bridge those scales?
It's an active area of research. Models like those HQMMs or even quantum neural networks, QNNs, are being considered for analyzing complex neuroimaging data or deep behavioral phenotyping data. The hope is that quantum parallelism might allow QNNs to learn patterns or correlations that are too subtle or complex for current classical deep learning methods to capture efficiently.
Linking brain activity patterns to specific cognitive states or behaviors more effectively.
That's the ambition. Finding those hidden structures in the incredibly complex data sets neuroscience generates.
This all sounds incredibly promising. Quantum computing poised to revolutionize biology. But there's always a but, isn't there? The sources mention a major bottleneck.
Yes. The big caveat, especially when dealing with the massive data sets common in genomics and neuroscience, is the call-RAM limitation.
Call-RAM, quantum random access memory. What's the issue?
The issue is that many of these powerful quantum algorithms we've talked about, especially those offering speed-ups on data analysis tasks, implicitly assume the existence of call-RAM.
And what would CROM allow you to do?
It would allow the quantum computer to access or query a large classical database, like a whole genome sequence, or a massive set of fMRI images in quantum superposition. Basically, load all the data points simultaneously into a quantum state to start the calculation.
Which sounds amazing, but
But building a functional, large-scale QuanRAM device is currently highly speculative. We don't really know how to do it reliably or practically yet. It's a major engineering challenge.
and without quorum.
Without quorum, trying to feed a huge classical dataset into a quantum algorithm piece by piece destroys the superposition. The very act of accessing the classical data causes decoherence. This means that for problems dominated by processing massive amounts of input data, current quantum computers often don't provide a speedup over classical methods.
Because the quantum advantage right now is mostly for problems with complex calculations, but maybe smaller input data sets.
Exactly. Or problems where the complexity lies in the quantum simulation itself, not just processing classical data. This QRAM limitation is why the hybrid classical quantum approaches we discussed earlier are so crucial right now. You use classical methods for the big data handling and focus the limited quantum resources on the most complex quantum parts of the calculation. Hashtag tag 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 outro.
Wow. Okay, so this deep dive has really taken us on a journey. We started with this, frankly, almost unbelievable idea of a mycelium quantum computer, this vision of biohardware, sustainable, maybe even self-repairing, using nature's own quantum dots.
And we saw how the detailed, meticulous science of mycoponics provided the crucial underpinning. Using advanced imaging and tracing, it revealed that nutrient transport in fungi isn't just simple chemistry. It seems to rely on fundamental quantum effects like tunneling and coherence.
Which then led us to how we can use quantum computation to understand and optimize these biological systems. Algorithms like VQE and QAOA are being developed to model protein folding, simulate molecular interactions, potentially leading to breakthroughs in sustainable agriculture or new bio-inspired technologies.
Yeah, and the whole picture painted by the sources is this intricate feedback loop. You have classical tools like MD simulations and neural networks interacting constantly with quantum tools like VQE, all being fed and validated by real-time data from experiments like mycoponics. It's this continuous cycle of measurement, simulation, and optimization.
It really highlights how understanding nature at its most efficient might require this incredibly sophisticated blend of classical and quantum approaches working together.
Which brings us to a final thought, something for you to maybe mull over. If the best way we can figure out how to understand and optimize these highly efficient natural systems, like fungal networks, is through this complex, adaptive, quantum classical computational loop.
Does that suggest that nature itself, at some fundamental level, might already be operating as a kind of vast, ongoing quantum classical computation, constantly optimizing, constantly adapting, using these very principles?